Great.
Yes. So our next speaker, we have Julie Hagelin and Tom Paragi. I think Julie's going to be doing most of the talking, as I understand, but Tom's going to be on as well. Both these folks are wildlife biologists for Alaska Department of Fish and Game. Julie specializes in small animals and Tom in habitat. They're going to be talking about little critters serving the purpose of inoculating soils and how that those interactions might look in forests. Julie, before I butcher anything too much more, I'm going to turn it over to you.
That sounds great. Thanks, Clay. And thanks to SAF for inviting Tom and me to give you this talk today. So my name's Julie Hagelin, and my co-author, Tom Paragi, and I were both from the Alaska Department of Fish and Game. And we want to talk with you about a frequently overlooked forest-wildlife relationship, which is highly relevant to reforestation, as well as forest resilience in a changing environment. And this has to do with the role of small mammals that disperse mycorrhizal fungal spores. And these fungi exhibit obligate associations with the roots of harvested trees, and it creates a symbiosis which is necessary for tree survival. So let me try to advance my slide. All right. So an important impetus for this talk actually came from a recent email that Jeremy Dowse sent to Tom and me, and it comes from this article from 2016 in the Northern Woodlands magazine. And this article actually asked an important question about forests in Minnesota, and that is, why do all conifer seedlings within a year or two die if they germinate in places that are away from adjacent forest? And in this case, it was old beaver meadows. And the answer lies in a critical forest-wildlife interaction that involves vole inputs of mycorrhizal fungi into the soils. So this is well known. It has a big impact on reforestation success in other regions of the continental US and in Canada. And Jeremy wondered if this might also transfer to places like interior Alaska. And if so, it would be quite relevant to regeneration, as well as timber harvest of mature stands, and also questions about carbon sequestration. So it turns out that Jeremy, if you're in the room, you asked just the right question, because this is something that Tom and I have been thinking about for a while. So what I'd like to do now is give you an overview of the structure of this talk. And I'm going to be talking about 3 different topics today. First, I want to review some forest-wildlife relationships or interactions that we think are important, that provide beneficial outcomes for both regeneration of timber in forests, as well as provides for resilience of forest stands. And we can talk about management opportunities and also why we should particularly care about this in places like the interior of Alaska. The next, I want to talk about 2 sister small mammal species, the southern redback bull and the northern redback bull, and what is known about their impacts to northern forests, like in Minnesota, the topic of that Northern Woodlands article, and also the redback bull, which is, the northern redback bull, which is the most abundant small mammal in terms of biomass in Alaska, and what's known about its role in fungi distribution. And finally, I want to present a roadmap for the future. As Jeremy suggested, Fish and Game also sees potential valuable opportunities for hypothesis testing and for understanding various forest treatments and quantifying both regeneration as well as wildlife responses. So let's first talk about these forest-wildlife relationships that Tom and I have been thinking about recently. The background to this is that in 2016, we were both part of a panel for the FERPA Reforestation Review with the Division of Forestry, and this caused us to uncover several key relationships, which we then later in 2020 expanded upon on the right-hand side of the slide as part of an ADF&G technical bulletin. This bulletin provides voluntary best management practices that managers can use to reap these beneficial outcomes that wildlife can provide for the forest by keeping just some basic principles in mind that maintain some key habitat features that many different wildlife species require. So first, let me give you a couple examples of what we feel are particularly important beneficial forest-wildlife interactions. And the first one I want to talk about focuses on bird-insect relationships. Many avian species, as I'm sure a lot of you know, are insectivores. And woodpeckers, for example, play a significant role in regulating and stabilizing populations of conifer bark beetles. So these are from the genus Dendroctonus and Ips. And this is evidenced by empirical observations, exclosures, modeling experiments, many of which are described in this Fate et al. Review article. So one management strategy is to maintain snags. These are important surfaces that that the woodpeckers use for feeding, but also for reproduction. And this maintains their beneficial impact on the forest. Other leaf gleaning birds, like warblers, also are effective predators of pests, including invasive insects. And here you have at the top a Townsend's warbler gleaning away at what I think are leaf miners in that photo, and also yellow warblers eating caterpillars. Now, I want to completely acknowledge the important role of parasitoids and pathogens in mitigating insect eruptions that exceed the feeding capacity of vertebrate predators. But I also want to highlight the role of birds in their ability to suppress and control herbivorous pests before an eruption begins. So vertebrates can delay and stabilize and avert epidemic level proportions before this happens, before the outbreak occurs, keep insects at low levels. And I even recently learned about some classic studies in the 1970s, which demonstrated the ability of birds that were actively feeding on an outbreak of European spruce sawfly to transport live pathogens, especially viruses, in their feces, which the birds then deposited on other trees that were infected by sawflies. So the birds themselves were actually acting as a vector and distributing pathogens during an outbreak. Now, whether that actually occurs here in boreal forest is completely testable, but it's currently unknown. So a second example that Tom and I have highlighted in our work involves the role of small mammals and mycorrhizal fungi relationships. And this is really important in Alaska. And actually, Clay, can you see the bottom of my screen, or is it grayed out?
I can.
You can see everything?
I can see it.
Okay,
yep.
Awesome. Great. That's wonderful. So this quote by Gary Larson, who's a UAF professor emeritus and a mycologist, really underscores the point that this is an important relationship in Alaska. And this was published in a Denali National Park scientific publication. So how does this work? Well, the spores of mycorrhizal fungi develop a symbiotic association with the roots of trees, which are shown here in this sort of magnified circles. And when the fungus forms a symbiotic relationship with the roots, it facilitates both nutrient and water uptake. And it's a relationship that's called ectomycorrhizal. It's on the outside of the roots as well, and it provides the roots with some protection from desiccation. So one common type of mycorrhizal fungus is called a hypogeous fungus. Sometimes these are called truffles or false truffles, and they're unique in that they've lost their ability to disperse spores through air currents. They create these subterranean fruiting bodies that are instead, the spores from those subterranean fruiting bodies are only dispersed through small mammal consumption. So in cross section, you can see this false truffle actually has spores inside. So I've put some truffles there underneath the soil. You can imagine along comes a little vole or a small mammal, It feeds on this ball of nutritious fungus, and then later its fecal pellets disperse and inoculate the soils with spores. So fecal pellets themselves, of course, are nutrient rich and they're beneficial for both the trees and the fungus. And this is a well-known, well-documented type of mutualistic relationship. But the cool thing is voles are not the only ones that do this. Lots of other small mammals do it as well. And like on the top branch here, this is a northern flying squirrel. Red squirrels down below it are also known to perform this service. And sort of as a piece of show and tell, I don't know if you guys can see this, it sort of looks like a moose pellet, but this is actually a hypogeous fungus that was dug up by a northern flying squirrel. It's about half the size of a fresh one. This one is quite desiccated. So they do exist. And this appears to be a universal relationship in almost every forest. So this obligate relationship with mycorrhizal fungi occurs across the genus Picea. So it's common in many conifers and hardwoods. And it even occurs in places like desert ecosystems, and even analogous systems to this have been documented in marsupial mammals in Australia. So to give you another sense of the sort of quantitative wow factor about how potentially important small mammal inoculation of soils actually is, this is the proportion of fungi from the digestive tract of over 30 small mammal taxa from Oregon coniferous forests. And what I've highlighted here is that 88% of these spores in the digestive tract are from hypogeous fungi, and 79% of those spores are known to have ectomycorrhizal associations with tree roots. So one management strategy is to maintain wildlife habitat that protects and provides cover for small mammals from predators, such as coarse woody debris. This provides some sort of visual cover, and small mammals can hide in it and not get eaten by predators. This will also promote small mammal inoculation of soils with ectomycorrhizal fungal spores. And another possibility is to maintain cavity trees, which are crucial for reproduction for squirrels. So an overriding goal in these two aforementioned scenarios that I've told you about and that are highlighted in our technical bulletin for managing timber and wildlife in forests relates to maintaining a diverse set of forest wildlife mediated linkages and functions that are not only helpful in that they can protect forests from insect herbivores and promote regeneration through fungal spores, but they protect, there's additional evidence that overall, maintaining a diverse set of forest wildlife interactions actually makes forests relatively more resilient to stressors and change. And it prevents forests, which are complex systems that are kind of in a dynamic balance, it protects them from going off on wild or unstable trajectories that more simplified natural systems are prone to doing. And this McCann article in Nature does a really good job of explaining the reasoning behind that. So our technical bulletin recommends how managers can do this at a stand and landscape scale in Boreal Alaska. So at a stand scale, maintaining late cereal features like cavity trees for squirrels provides habitat for those insectivorous birds I told you about, which help to avoid less predictable or destabilizing outcomes by keeping the risk of deleterious or invading insect species in check. Cavity trees also provide habitat for key predators like owls and other raptors, and even marten, which keep eruptions of herbivorous mammals in check. And late cereal habitat is important for lynx, which keeps the hare populations down and thereby reduces the risk of girdling trees, which are at greater risk when the hare population cycle is at a high. So also at a stand scale, maintaining coarse woody debris creates small mammal cover. It allows for forest regeneration through the distribution of mycorrhizal fungi. and it creates resilience of larger trees against stressors such as climate change. So you have more and faster regeneration and greater resilience of mature trees. And this combination makes for greater potential for timber harvest as well as carbon banking. At a landscape scale, the goal is to retain islands of diverse sets of habitats and some connectivity between them. So you get a patchwork of landscape in different habitat types and ages, and this maintains a diverse set of forest wildlife linkages. And our bulletin even describes ways of mimicking large natural disturbances similar to fire. And this would include large harvested areas, but with enough habitat islands within them that contain habitat features so that both wildlife and timber can repopulate, regenerate, and continue forward as a relatively stable dynamic system. So this really provides opportunities for testable hypotheses and management strategies that we can work on together. So another reason why we should care about this is that Alaska's boreal forests are actually quite special due to our history of unglaciated areas known as glacial refugia. And Roberts and Hamann identified two important refugia within the boreal forest when they created heat maps across Western North America. And they looked at the species richness, so the number of different tree species out of 22 Western trees. And this far north, our two refugia here are indicated in red shading, and they stand out as having high tree species richness. But it turns out that within these tree populations, these refugia actually harbor even greater numbers of greater amounts of genetic diversity in terms of the numbers of alleles. So this stands out across Alaska, and it also stands out as an important area in North America, Western North America. And the reasoning here is that this genetic diversity has accumulated and been maintained because in other places of Alaska and elsewhere, entire regions of trees were wiped out due to glaciers. But here it's had time to collect in glacier-free areas. So what's a way of more efficiently regenerating our genetically unique and special Alaska boreal forests? What I'd like to do is zoom in and talk about in more depth, more about the biology of what's known of these two sister species of redback voles with regard to mycorrhizal spore dispersal. So the southern redback vole and the northern redback vole that we have here in Alaska look pretty much the same, although the northern, I think, looks a little bit cuter because he's eating blueberries. Both of these species are very common. They make up the biggest proportion of small mammal biomass in many of the habitats where they occur. They're considered a keystone species for many predators. They're the basis of the food chain. And these predators that feed on voles also regulate other pests like hares, which are prone to girdling trees. So as their name suggests, the southern redback vole has a more southerly distribution, and the northern redback vole, so southern redbacks are in northern continental US and parts of Canada. The northern redback vole has a more northerly distribution. They're also found in boreal parts of Europe and farther to the east. Southern redbacks have been more extensively studied in the Pacific Northwest and BC and the upper Midwest, where I mentioned that beaver meadow problem. In Minnesota, after 70 years, no conifers have invaded beaver meadows, just like this picture. And the big question is why? And research on this topic led to some really interesting discoveries. So the tree genus Picea, including these spruce seedlings that you see here, are obligately dependent on ectomycorrhizal fungi. And John Pachtel, who's the professor who wrote this article that Jeremy sent us, had a grad student who grew seedlings in beaver meadow soil and in forest soil. And after quite a few weeks, none of the seedlings grown in meadow soil formed root mycorrhizae, and all of them grown in meadow soil died over time. But 100% of the seedlings grown in forest soil formed mycorrhizae. And so there was something special about that. 100% of those seedlings survived. So this student did a test treatment where he artificially inoculated the soils with vole pellets. So they effectively added fungal spores to the soil with vole pellets. And 30% of the seedlings survived with May vole pellets collected in May, whereas even more survived with August vole pellets. And August is the time of year when voles are consuming even more hypogeous fungi in their diet. So if the Division of Forestry were ever to get a seedling propagation activities going locally, I'd strongly recommend obtaining a supply of local vole pellets, especially during the fall. So treatment of these meadow soils effectively experimentally rescued the seedlings that otherwise suffered from a lack of mycorrhizal spores. Now, there's of course some nitrogen and phosphorus in vole pellets, but other investigations have shown that the amount that's present in the vole pellet is far less concentrated than the needs of the seedling. So it wasn't just fertilizer that was added that made a difference, it was the spores. And dominant small mammals in these beaver meadows are also known as a meadow vole. And these meadow voles are quite aggressive and they keep out redback voles. And also meadow voles do not consume hardly any hypogeous fungi. So there's no opportunities for the soils to get inoculated. So from a regeneration standpoint, the seedlings in the forest soil actually grew twice as large as those in the untreated meadow soils. And those in the meadow soil that received vole pellets grew 20 to 30% larger than those that grew in and ultimately died in the untreated meadow soil alone. So you can imagine this Minnesota meadow effect is somewhat analogous to the landscape after a disturbance, including tree harvest, just like this photo. So as Gary Larson points out, disturbance itself can damage the spore inoculum in soils. And post-disturbance, there's even less habitat for voles. So that provides for an additional loss of spores in the soil. So the question becomes, how do we get this back? And there's two things to consider here, which are known for southern redback voles. First is the response to green tree retention, different harvest treatments. And the second is response to different treatments of coarse woody debris. So let's go through each one of these. Green tree retention has been studied in the Pacific Northwest on southern redbacks, and this depends upon the amount and the configuration of green tree retention. So you can imagine different harvest practices which might retain aggregate islands of forest in differing amounts, or you could have green tree retention simply as dispersed trees evenly across the landscape. And for each of these two treatment types, you can imagine different levels of retention. So here I've shown for the aggregated treatment only, because this paper only provided this kind of figure. But here you can see at one end of the spectrum, the maximum is 100% aggregation, that's uncut forest, which is shown right here. And at the other extreme, you have only 15% retention. And these little black blobs equate to these aggregated green tree, smaller bits of aggregated trees and smaller stands. So what I'm not showing you, but you can imagine, would be the dispersed treatment types. These would be different densities of sort of evenly dispersed trees, different densities or different stippling patterns representing different retention levels. So let's think about what happens under these different conditions. Well, if you look at vole pellets, you get greater amounts of spores and pellets from the aggregated treatments over the dispersed treatments. And it's kind of unclear why this is happening. Either hypogeous fungi may be less present in the dispersed treatment, so there's less opportunity for voles to feed. But another possibility is that voles are less willing to venture into open habitats due to predator danger. They have no way to take cover. So the dispersed treatments didn't work as well. So let's just focus on what happened in the aggregated treatments. Within the aggregated treatments, pellets from habitats with the least levels of green tree retention had fewer spores. But those with that had 40% or more green tree retention had more spores present in the fecal pellets, and this was the most similar to the uncut forest control. So having green tree retention at 40% or more seems to be a good way of maintaining voles that will inoculate soils in a similar way to what you have in uncut forest, just based on spore counts in fecal pellets. What about coarse woody debris, which is known to be important vole habitat? Now, multiple studies indicate that CWD is very helpful to have over no CWD at all. It provides this really important small mammal cover. It retains moisture better for small mammals and possibly also the trees. And it's also good for hypogeous fungi. So here's a dispersed coarse woody debris treatment, and Craig et al. Found that at levels greater than 75 meters cubed per hectare, this is good for redback voles, southern redback voles in southern British Columbia. But you can imagine you could have different types of treatments of coarse woody debris. You could have this dispersed treatment, or you could have a more organized treatment where you create windrows and you connect those windrows to uncut forest. So these are more similar to aggregated dispersion of coarse woody debris within the harvest area. And it's importantly connected to either your aggregated green tree retention islands or uncut forest. So this sort of study is exactly what Sullivan did in 2017 at the University of British Columbia. And he concluded that how coarse woody debris is provided actually makes a difference. So let me walk you through this slide. This is over 5 years of data collection. On the x-axis, you have time, and the number of voles are measured over many months each year. Voles go through a natural cycle where they have reproduction in the summertime, and then their numbers kind of fall over the winter, and then they're measured again. And what I want you to notice is, so more voles is better. What I want you to notice is that the dispersed treatment sort of perpetually resulted in low vole numbers, and that's not really what we want. But if you look at the darker lines here, especially the dark triangles, is you see this is the windrow treatment that's connected to uncut forest and creates these kind of linear piles of wood pathways for small mammals. That was quite important in terms of getting lots of small mammals into the harvested area. And it was on par, quite high here, on par with the uncut forest areas in terms of the number of voles that it brought in to the harvested area. So I want to especially acknowledge here that windrows contain larger diameter green debris, which are prone to IPS outbreaks. So if we were to ever field test a treatment like this, we would really need to do this in consultation with a forest health specialist. But the authors of this paper emphasize something rather important, I think. So it's not just windrows, but even in larger scale disturbed areas where windrows maybe aren't practical to build after a large scale harvest or after a large insect outbreak or a big fire or salvage log operations. They emphasize that creating some kind of linear features with woody debris that connects to existing forest is really key to getting this beneficial impact from small mammals to occur. So I've mentioned that small mammal distribution of fungal spores is crucial to many species of forest timber and to the management of this renewable resource, including in Alaska. But can we transfer some of these lessons learned from southern redback voles to our species, the northern redback vole here in Alaska? Oh, and I should mention here, before I go to the next slide. So we've got windrows connected to the forest, had more voles, did better than those with dispersed coarse woody debris. But another really important conclusion that they found was that these windrows also had greater small mammal diversity. And we know that many small mammals distribute mycorrhizal fungal spores, not just redback voles. So this is an extra payoff of having higher levels of small mammal diversity. We want that. And the windrows were able to provide that as well. So let's talk now more about our species, the northern redback vole, and see if these same principles translate to Boreal Alaska. And there's 3 important facts that we don't know as much about northern redback voles, but we do know a few things that point to kind of an exciting direction. First of all, mycorrhizal fungi are really important to regeneration of spruce, birch, and aspen in Boreal Alaska. It matters. Their stem diameter and shoot mass are positively associated with mycorrhizal fungi. And we know that this inoculum of spores must be present in the soil. Secondly, these hyphageous fungi that voles eat are known to occur in mature white spruce and deciduous stands. Some, like this picture here, you've got a cross section of a hyphageous truffle. Some of these, like this one, occurred in mature white spruce just down the road in the Bonanza Creek Experimental Forest. So we know these exist here in Fairbanks, and it's likely that they exist sort of throughout boreal forests in Alaska. This particular hyphageous fungus was taken from a 135-year-old mature white spruce. And finally, a paper that I was involved with showed that northern redback voles have the highest percent of fungi in their diet compared to other small mammals. So they make up the greatest amount of small mammal biomass in our state. So they're likely to be a key driver of mycorrhizal fungal spores and fungal dispersal. So even experts on southern redback voles like Thomas Sullivan at the University of British Columbia agrees that the relationship with mycorrhizal fungi could be similar between these 2 sister species. They both belong to the genus Clethrionomus. And it's not just fungi that they're distributing. Voles aerate soils at a massive scale in the forest year-round through their tunneling behavior. And not only that, but here in interior Alaska, we can benefit from long-term small mammal data in Denali. So we actually know that our redback voles show 2 to 3 year population cycles. So you might expect that during a year of a vole high, we might expect voles to have the greatest inoculation impact of soils with the mycorrhizal fungi. So it'd be good to design experiments that straddle or are able to capture a vole high year. And we know when that's going to happen. So we know when to put experiments on the ground. So Tom and I think there are real research opportunities for ADF&G and for forestry to partner in some research like this. And the big question now is, well, how would we do this? And I think I see this happening in kind of 2 steps. So before you sink a ton of money into field treatments, I would strongly recommend an important initial step, and that is to verify that northern redback voles actually distribute mycorrhizal fungal spores of harvested tree species. And how you can do this is pretty simple. You simply look at the DNA present in the diet of vole pellets. And we've already done this for another small mammal called the northern bog lemming. And here on the screen, you've got listed different families and different genera of taxa in the diet, and the ones listed in brown are fungi, and the bars provide an indication of how many DNA reads you got in your sample, so that you can use this to estimate the relative abundance of each dietary item, so you know how important it is. And we already have a sense of at least a few mycorrhizal fungi that are important to white spruce and birch, and there's others that are available in the literature. I uncovered a study from the Kenai that created assays that you can run on tree roots to determine the DNA of the ectomycorrhizae from the tree itself. You can also run tests of the soil to use as a comparison. So once we've established that, and I'm not a betting person, but I'm willing to bet that we would establish that pretty quickly, the next step is to test the importance of redback vole inoculation in the field. And we would want to time this during a vole high year so that we would capture the vole's maximum impact. And we'd focus on 2 treatments and take advantage of settings that have worked optimally based on other southern redback vole studies. So 40% green tree retention, compare what happens under that circumstance relative to an uncut forest control. So you can also consider other treatments, not just green tree retention, but other treatments that involve coarse woody debris, like connecting windrows to uncut forest. Or we might want to create some linear debris features that connect to aggregated patches of green tree retention. And the response variables that we might want to measure here could be regeneration rate or seedling stem diameter or mass. We could also imagine measuring wildlife responses. So compare the spores in pellets among these different treatments to figure out how to maximize spore availability. Also look at the number of voles present or the small mammal diversity. Remember, more is better. Many of these species distribute mycorrhizal spores. So to kind of wrap up here, I want to provide you a sort of a simplified view of the forest cycle. And this idea that I'm going to be talking about comes from Glenn Juday at UAF, who recently presented some of this thinking at a Wildlife Society meeting in Fairbanks just a couple of weeks ago. So we all know that after a disturbance, that young trees establish, they grow into mature stands, and then eventually enter this late cereal phase. So small mammals provide one of the only ways that links older forest to younger forest that I can think of. This is a critical wildlife linkage. It provides this key connection between different phases of the forest cycle. And given the obligate nature of spruce and other species that require mycorrhizal fungi, regeneration just wouldn't be possible without vole inoculation of soils. And we've discovered that soil inoculation can vary with different forest practices, which can be treated as testable hypotheses. And the key is to find out what works best in Boreal Alaska. But also in the process of retaining key habitat features for mycorrhizal dispersers, not just voles, but other key species like flying squirrels or red squirrels, by retaining cavity trees for nesting and aggregated islands of habitat connected to uncut forest, you also pull along other benefits. And here are some of the few examples that we've talked about today. The cavity trees in unharvested patches provide habitat for avian insectivores that create a relatively stabilizing impact by keeping insect populations in check. And these cavities also maintain reproductive habitat for other top predators like owls or marten that promote more checks on herbivores that can damage seedlings. And islands of uncut habitat also provide habitat for lynx that can keep hare numbers down and keep them from girdling trees. And finally, if we focus on the younger phase of the forest cycle, by maintaining habitat for moose, moose can come into these early successional stages and forage and clear away the encroachment of hardwoods and willows and create openings or patches of sunlight where conifer seedlings can successfully mature. So given all this complexity, I have 3 simple take-homes for you today. The first is that forest-wildlife relationships are important. They matter. And the lessons that we've learned from southern redback voles are likely applicable to our northern redback vole here. This is critical inoculation of soils with mycorrhizal fungi. And these lessons for voles are not only applicable to harvested areas, but occur after any kind of disturbance. We can apply them to regeneration post-fire or how to recover after an insect outbreak faster. And finally, these mycorrhizae are crucial to many forest timber species and to the management of our renewable resources, and they're mediated by small mammals. So these small mammals also have an important consequence for forest management. So that's all I had for you today. we're gladly, if there's time, take any questions. But most importantly, Tom and I would be grateful for some feedback. We want to know, do you all think this is a worthwhile idea? Are there other things that we should be considering? And please feel free to email us if you think of other aspects later on. So thanks a lot.
Thanks, Julie. Are there questions?
Oops.
Hey, Julie, this is Jeremy Dowse. Really good presentation. Thanks for being willing to do it. The thing that came up in my mind when you were talking about kind of the layout of the units, the harvest units, is like, how would this affect the logger, and how can they implement a prescription like this? And it seems to me, I mean, I guess this is more of a comment, but it seems to me like this is all doable. Like the aggregated retention of the stand versus the dispersed, that totally makes sense from a logging perspective. And at least in my experience, it's been easier to work with aggregated like retention patches in a unit versus like a seed tree where a lot of the retained trees get damaged through the hauling or through yarding. And then the aggregator or the windrow piles, I was thinking that that might be a little bit more tricky right now. At least on state timber sales, we're doing mostly whole tree yarding to a landing, which includes all of the branches and the tops as well. But you know, the loggers in some cases want to process the tree at the stump and then just yard the stem to the landing. But then you've got more of a dispersed material throughout the unit rather than in windrows. But then, you know, you could potentially pile it up into those connective sections between retained forest through the scarification process. So anyways, I just think that this is all like doable with our existing harvest systems that we have, at least in the interior. So
that's great to hear. Thank you.
Okay, we got one over here. Mitch?
Hi, about 20 years ago at an SAF meeting, Andy Youngblood and Gary Larson were here, and they presented, Gary had a grad student that gave a presentation on redback voles, and Youngblood was giving a presentation on the effects or the effectiveness of varied site prep treatments in the Copper River drainage. And one of the things that Youngblood said, which he said there was a definite correlation between the amount of large coarse woody debris, and not just the survival, but the height of the tree heights, seedling heights. And Gary pointed out at that point that you should look into the research that his grad student was doing, which was the populations of redback voles as it correlated to how much coarse woody debris was on. And it generated a wonderful discussion about how the redback vole essentially is playing a keystone place in this whole regeneration of our forest and the importance of coarse woody debris. So I'm really glad you did this research, because that's
thank you.
It should continue.
As you can tell, it's pretty exciting. I love it. But I'll look further. I've been trying to reach out to Gary and get more information now that he's retired. I think he lives in Seattle. So some of these past activities that he and his students have done, I haven't been super successful in finding. And so I don't know if they're published or not, but they're really critical, and it would provide us just even more insights on how to move forward. Thank you.
Hey, Julie, we can do one more quick one.
Okay.
Hey, Julie, Sue Rodman here.
Hi, Sue.
Thanks. That was awesome. Would you be willing to drop the link for your technical report in the chat so folks can find it? Thanks.
I can do that. Great idea.
Doug?
Okay, I will stop share.
Hi, Julie. Hopefully this is a quick question. So the first thing I was going to say is great presentation. I don't know if you know of similar work on windrows that's been done from the John Prince Research Forest that's associated with the University of Northern British Columbia. That might be of interest to you. It's not focused on voles, it's more to do with fisher, but that, you know, it might be a sort of interlinked thing there. So, but my question is, as foresters, we deal with a lot of competing values that we have to balance. And of course, from a scientific research perspective, you're looking at this without values, you're just looking at the data. But of course, 40% retention is quite a paradigm shift. And I'm wondering what you think are going to be the biggest points of resistance trying to promote this idea, and maybe some ways to address those points of resistance. And maybe you haven't thought about it. And if you don't know, that's fine, because that'll make my question nice and quick.
Yeah, I think the key here is really to have discussion going with foresters and figure out what it is that they need. And I think that many of these publications have shown that there are ways of, maybe you're not going to get inoculation levels back to 100% under certain forest treatments, but there are ways to bring it back so it's relatively better than doing nothing. And that will improve regeneration. I don't know, Tom, you've got an idea?
Well, I think one of the key things is that in our bulletin, we talked about the idea of actually going to larger timber sale unit sizes. It kind of surprised some of our colleagues, but what we pointed out is typically from an operational standpoint, you're trying to provide a desired amount of volume for an operator in an area. And as Jeremy said, you can aggregate that much easier than doing it dispersed. So what might have been a 30 or 40 acre timber sale area, you might double that or some higher proportion so that you can still get out the volume within your managed area there. But overall, you're still maintaining these habitat relationships and linkages that Julie was describing. And that would be the paradigm shift for some members of the public and some biologists. But if you look at mimicking the size of burn scars in the boreal forest, they're huge. They're a couple orders of magnitude larger than most timber sales easily.
The most important thing is retaining some kind of islands of habitat within there so that you can repopulate these areas and inoculate the soils and have those ecosystem services of wildlife still available. But our boreal forests are used to large scale disturbances. So this seems to be kind of ecologically a logical place to test that sort of thing, large scale harvest.
And there has been research out of Canada showing that the larger the scale of burns, the higher the proportion of unburned within it as burns occur over time. So bigger burns typically have larger unburned patches. Not all of them, obviously, but that's kind of a pattern that's emerged in the research.
Well, thank you guys. I think we probably have more questions, but we'll have to get you outside of the meeting. Thanks for showing up, and great presentation.